So very cool to be here making this introduction for a friend of mine who I've known for an awful long time. This is his second seminar at Moss Landing, and he just reminded me, well, his first one was in 2005, and I'll tell you a little bit about that in a sec, but it was back when I actually had a keg in my lab, and we used to wheel it down the hall for every seminar. It sat over there. So he was just remarking how things have changed, where we no longer have the fresh tap sitting in the seminar room. So we'll make up for that at dinner. So this is Matt Bracken. Matt, I've known him forever. He was actually a postdoc on my second NSF grant I got. So I got my PhD in 2000. I got here because these guys hired me in 2002. Yep. And I got an NSF grant with my collaborator at UC Davis, which was Jay Stakowitz. And we had a 4-year grant to study the biodiversity of seaweed systems in the intertidal at Bodega. And Matt had just come on as Jay's postdoc. So I got to work with Matt for about 1.5 years on that. And then about halfway into that postdoc, he kind of fledged out, came down here and started telling us about his own stuff. And then he went on to have a great career. So he was here when we were just starting the new round of beer pigs. And so I said, hey, can you give me that was a long time ago. And I've seen him in meetings and we interact, a decent amount. And I said, give me a CV. So, these guys that don't have full professorship yet send you a whole CV, which is every single presentation they've ever given. And then I started going through and I thought, well, that's pretty cool. You know what? In 2017, Matt was the Adopt a Scientist pen pal for an elementary student, which is pretty cool, I gotta admit. So Matt got his bachelor's up at University of Puget Sound a while ago in biology. He then went to Oregon State and worked for Jane Lubchenco and Bruce Mengi, got his PhD there. And I think that was in 2003. That was right after I got here. He showed up at Davis to be a postdoc, first in Jay Stakowitz's lab, and later he was working with Susan Williams out at Bodega Marine Lab. And then he got his first faculty position out at Northeastern University, and I think it's in 2007. And is that Shoals? Which marine lab were you primarily associated with?
Northeastern University Marine Science Center.
Okay, so he's out on the coast doing there. It's a little harder when you work on algal systems on the East Coast because they just don't have very many species of algae. So it's obviously much more exciting when you're working on algal and invertebrate systems in the intertidal to come back to the U.S., to California. And in 2014, he got hired as an associate professor at UC Irvine, Southern California, which is where he's been. He's got 3 current grants going right now, one of which is looking specifically at functioning in the intertidal, which is what we're going to hear about today. He's got a whopping 54 papers, of which I'm an author on 3 of them, not the first author on any of them. He's always higher on the list of authorship than I am on those papers. But it's been a lot of fun. So Matt is back on this coast, which is where he really started everything. He's a functional ecologist, which is really trying to look at how these systems work. He tends to spend a decent amount of time up in the intertidal, where there's lots of invertebrates and lots of seaweeds. Spends a decent amount of time working on energy and nutrition and stuff like that. Is down in Southern California, where everything's a little wonky right now, but has a perspective that spans from Alaska, which I'm sure he'd love to tell you guys about, all the way down into warmer climates. So it's a very broad background, very diverse applications, and again, a young faculty member down at the UC system, which we're glad to see here after 15 years was the last time he was here, 14 years. So we're going to let Matt start his talk on the unanticipated roles of consumers in mediating the functioning of marine ecosystems. Thanks for coming, Matt.
So it's a real pleasure to be here. As Mike said, it's been a while, well over a decade since I was on the Moss Landing Marine Lab campus. So it was really cool to see all the changes that have happened since then.
I only had 2 kids then.
Yeah. I had no kids then, and now I have 2. The crazy thing is one is a teenager, so that kind of tells you about the passage of time. And to visit the seaweed farm, which was especially cool. Today what I want to talk to you about is the roles that consumers play in mediating marine ecosystem functioning, but not in a traditional way. So I don't want to focus on their top-down roles, but actually their roles in mediating bottom-up processes. Before I get started, though, I want to thank people, funding agencies, places that have provided access for this research, and what I particularly want to highlight. So everyone whose name has an asterisk behind it is an undergrad who's worked on my research with me, and I really try to involve undergraduate researchers. So here are This is sort of the gallery of undergrads doing work, and highlight their role in this process. Okay. So one of the central organizing themes of my research program is understanding causes and consequences of biodiversity change. And one of the reasons for that is that, as I'm sure all of you know, humans are changing the diversity of life on Earth. And so we've seen a whole bunch of recent papers, like this one by Naeem and colleagues, highlighting the fact that we now live in an age of extinction, or thinking about the emerging mass extinction in the oceans, or that we may in fact be entering the 6th mass extinction. And this all sounds like doom and gloom and hyperbole, but maybe it's not. So this is a figure that I put together from 2 different sources. I took data on past extinction rates from a paper published in Journal of Paleontology by Sepkoski and Raup. And that's all of these data that extend back about 600 million years. And then I combined them with some more recent data collected by Stuart Pym and colleagues that actually looked at observed extinctions over the last 100 years or so. And this is the rate of extinction that we've observed over the last 100 years, relative to what has occurred at any time that we can see over the history of the planet. And while I fully acknowledge that there are difficulties in combining fossil data sets with what we've actually observed, if this rate is anywhere near real, in this uptick right here, then natural systems are in real trouble. And the reason for that is what we as humans are doing to the planet. In marine systems, we're overfishing. Climate change is having big impacts. Terrestrial systems, a lot of it's based on land use changes. Invasive species are a major driver. Here's an example from some of my own work highlighting the role of an invasive seaweed species. So this is an example of being in the right place at the right time. So I had a whole cohort of well-trained marine botanists who were diving in this area. And they said, wait a minute, this seaweed species isn't supposed to be here. This was back when I was doing work in New England. And we discovered that this seaweed here, this red fluffy thing, is actually at the time we were calling it Heterosiphonia japonica. It's now been reclassified as Daisy Siphonia. And you can see that as the biomass of that invader increased in the plots that we were surveying, you see a corresponding decline in seaweed species diversity. So it's really driving these species locally extinct. And as humans are altering the diversity of life on Earth, they're consequently altering how biological systems function. So if we go back to this paper that I highlighted at the beginning about the age of extinction, it's actually really about the functions of biological diversity in an age of extinction. And a more recent paper by Greta Peckel and colleagues highlights biodiversity redistribution under climate change and the impacts of that biodiversity change on ecosystems and human well-being. So if you look again at this paper by Shahid Naeem, Emmett Duffy, and Erica Zavaleta, we can see sort of how these changes are manifesting themselves. So I particularly want you to focus on this area right here. And what you see is in a variety of systems, so this is forests being converted into farmland, savannas being converted into grazing land, or marine systems that are being overfished. And each of these systems is mediating important biogeochemical processes that operate at the scale even of the entire biosphere. And those processes are deeply tied to the roles that species are playing in these ecosystems. So as species are lost or systems are transformed, we're seeing changes in these sorts of processes. Here again is an example from my own work. So after I set up this experiment with Mike and with Jay Stakowitz at Bodega Marine Lab, which is focusing on what's going on at a single site, I made the, I guess, audacious decision to try to replicate that same experiment on the East Coast. It was easier because diversity isn't as high, but I did it at 3 sites in 3 regions spanning 500 kilometers of coastline. So I took the Bodega experiment and I made it bigger and bigger, right? So what I just want to highlight here is that we see patterns like this emerging, where as species richness increases, here on the x-axis, we see corresponding increases in seaweed cover. And this effect is consistent along 500 kilometers of coastline, and we did the actual experiments to establish that this is a causal relationship. So as diversity increases, we see that systems are more productive. So which species are at greatest risk of extinction? This is a really cool comparison. So this is a paper that was published by Jonathan Payne and colleagues highlighting ecological selectivity of this mass extinction, particularly in marine habitats. And what you can see is on the x-axis of these figures, this is time. On the left-hand side, this is going back 60 million years, so into the Paleocene. The right-hand side actually shows the 5 big mass extinction events over the history of the planet. And then M is modern, so that's the modern extinction that we're currently experiencing. And what you can see is that relative to past extinction events and past extinctions, either over the last 60 million years or over the entire history of life on Earth, the current extinctions are focusing disproportionately on larger organisms and more mobile organisms. So relative to the past, large and mobile species are disproportionately going extinct, and these are consumers. So in my talk today, I particularly want to think about what are the roles of consumers in marine ecosystems, and what are the consequences of the loss of these consumers for how these systems function. So a traditional perspective on this would be a trophic context. And this is I'm sure everyone here in this room has seen a figure that looks a lot like this one. But it's a good place to start. So usually you'd think about consumers eating their prey, right? So you have things like sea otters eating urchins eating kelp. And historically, and now as otters are reemerging as important consumers in these systems, otters enhance kelp by keeping urchins in check, right? So this is a well-known, well-described indirect effect. But then historically what happened on the West Coast is the otters were hunted to near extinction, so they were lost. Urchins increased in abundance and kelp declined. So the sea urchins decimated the kelp. So this is a really top-down focus on what the loss of consumers can do to how marine systems work. And what I want to introduce you to is some other possibilities, some other important roles that consumers play as we try to answer this question, what are the consequences of the loss of consumers for the functioning of marine ecosystems? I actually think it's a really fitting place here at Moss Landing to present you with this view. I did a lot of work over the past few years at the Rancho Marino Reserve on the Central Coast, where Don Canestro helped me out with a lot of my research. And so this is where I'm standing at Rancho Marino, looking out to sea. And then I look down at my feet and see this right here. So like trying to focus in on what's going on in that system at the local scale. And so you can start looking at the organisms that live there and try to put together a food web, thinking about the possible relationships between them. So here you have the starfish. If you look closely, you can see some snails like tegula in here, seaweeds, surf grass. You know there are microalgae in this system, nutrients affecting it from the bottom up. Now I want to zoom a little more closely into this system and focus on this subweb right here, because this, more than anything, is what we see in Southern California. A local intertidal food web where you have these snails. They're mostly eating microalgae. There are nutrients affecting the system from the bottom up. And think about the roles of the consumers in that particular subweb. Now, one of the things that I noticed when I moved to Southern California is that there aren't a lot of macroalgae, especially high on the shore, but there are a ton of grazers. So you have things like this owl limpet, and you can actually see, if you look closely, you see these grazing scars that's left all over the rock as it's been grazing diatoms. So it's scraping with its radula. It's got these nice little flat teeth on its radula that it's using to scrape away these microalgae. So if you look at traditional perspectives on the interactions in this subweb, you think about top-down versus bottom-up processes. So from the top down, herbivores are eating algae. This is a picture taken by one of my undergrads, Alex Badton, who split these. We were using tiles as a substrate for algal recruitment and growth. And here there are snails on one side and no snails on the other side. And you can see this dense algal bloom over here and no algae over here where the snails are eating those algae, right? So strong top-down effects. And there are also strong bottom-up effects. So this is actually a picture taken by a colleague of mine, Carol Thornburg, looking at an estuarine system. And you can see the algae that are blooming there associated with high nutrient inputs coming down that estuary, so affecting the system from the bottom up. But consumers also can shape nutrient availability in marine systems. So here's an example, and I'll spend more time talking about this in a couple minutes. But this is just looking at a bunch of the different grazer species that are characteristic of tide pools in Southern California and their ammonium excretion rates. So they differ in terms of their contributions of nitrogen into the system, but all of them are recycling nutrients and changing the availability of those nutrients at the local scale. And so there's a possibility that in addition to their top-down effects, negative effects on algae, they could also be affecting nutrient availability and altering the system from the bottom up as well. So that's what I really want to focus on here, are these sort of unanticipated effects or non-traditional effects of consumers on algal growth. And the way I want to approach that is first let you know, talk to you about how consumers can in fact enhance algal growth. Then a little bit about methodology. How do we partition the consumptive and facilitative effects of grazers on algae? Discuss using some of those designs, the importance of community composition and mediating these interactions, and then take those interactions into the field and say, well, what actually happens in a natural system when we try to tease apart these top-down roles of grazers versus their bottom-up effects in the system? Okay, so let's start with this first one. What do consumers do to algal growth? And again, I'm going to return to this example. So this is just highlighting how consumers excrete ammonium as a waste product, thereby affecting its availability in these systems. So here you have a variety of different grazers, littorine snails, limpets, chitons, and turban snails, all excreting ammonium. So these are their per biomass ammonium excretion rates in micromoles per hour per gram. You can see that turban snails on a per gram basis are actually the most effective excretors of ammonium. And one thing I want to highlight is that this is a pretty diverse herbivore assemblage, and that's one thing that really struck me when I moved to Southern California, is looking at these tide pools. It's like, wow, there aren't very many algae in here, but there are a lot of different herbivores. And I also want to highlight the fact that these two undergrads, Alex Badton and Jill Oates, headed up a bunch of this research. Okay, so I've modified this now, so we have littorine snails, limpets, chitons, turban snails. They're all both affecting algal abundance from the top down, but they're also mediating nutrient abundance from the bottom up. And that ends up being really important. So Southern California tide pools, so this is once I moved back to California, they have a bunch of different species of molluscan grazers. And we're interested in how they can collectively enhance algal growth and productivity via the ammonium that they're excreting at the local scale. So it's pretty easy to count them. You can estimate total biomass. Littorine snails, here on the top panel you see abundance. They're the most abundant in terms of their numerical abundance. But despite their slightly lower abundances, here you can see the relative abundance of turban snails. They're bigger, so they actually have the greatest biomass of these different grazer groups. So here you have the contributions of these different species to total biomass and also to the predicted ammonium accumulation rates in these tide pools. What we really wanted to figure out is, could we just go out and count them and then predict how much ammonium each of these different contributors was excreting in these tide pools? So here you can see their excretion rates in white, and then how much they're contributing to biomass in gray. And these are the percent contribution to total excretion, ammonium accumulation rates, and total biomass in these tide pools. And then in the bottom panel, you can see the difference. So this is the difference between the excretion contribution and the biomass contribution. So littorines are actually, relative to their biomass, are contributing very little ammonium, whereas turban snails are disproportionately contributing to ammonium accumulation rates in these pools. And the other two groups, limpets and chitons, are somewhere in the middle. So we wanted then to say, well, can we go into a tide pool in the field and predict ammonium accumulation rates and relate that to accounts of these different herbivores? So we measured ammonium accumulation rates in the field. And here's what you can see that here. So this is over time. This is ammonium concentrations. And then we said, are these rates predicted by ammonium excretion rates, or are they simply predicted by the total biomass of herbivores in these pools? And so here you can see those comparisons. So on the top panel, you see predicted ammonium excretion rates, and this is the actual change in ammonium concentrations observed in tide pools. And then this is herbivore biomass in that same comparison. So the y-axis is what we actually measured in the field, and the x-axis is what we predicted in the top panel based on knowing the ammonium excretion rates of these different types of herbivores. The bottom is simply if we said, well, it scales just with biomass. Turns out that biomass in and of itself is not a good predictor. You really need to know the identity of these different species or these different grazer groups to be able to predict the change in ammonium concentrations over time. And the other interesting thing is this is the one-to-one line. So this is what we would expect in terms of ammonium accumulation rates. But these values are lower than that, and that indicates a substantial amount of ammonium being taken up by the algae in these tide pools. All right, so it's clear that consumers are affecting nitrogen availability. The second question is, how does it affect algal growth? And to answer this one, at least initially, I'm going to take you back to work I did as part of my PhD. I've been working on in tide pools for a really long time now, Mike. So this seaweed species, this is Neorotamella oregona, and it commonly grows on mussel beds. Here you can actually see it growing on a live mussel, and I did experiments where I dissected the tissue out of mussels and observed their growth rates. Over time, this is percent growth where mussels are present here and where mussels are absent. So they grow a lot more where the mussels are present. And in fact, I was able in this experiment to relate ammonium loading associated with these consumers to nitrogen assimilation rates by the seaweeds, and you can see that they're coupled very strongly. So ammonium provided by the mussels is assimilated by the seaweeds. Consumers are clearly contributing nitrogen to these algae, and they're converting it into growth. But mussels don't actually eat seaweeds, and this is something that for many, many years I really wanted to tackle, and I finally came back and circled back around to answering some of these questions. Specifically, can herbivore-mediated nutrient inputs have similar sorts of effects? So in the mussel system, this makes sense. The mussels are actually getting their food from outside the system. They're acting like a conduit, filter feeding on stuff in the nearshore ocean, and then converting that material as a metabolic waste product into locally available nitrogen that then enhances seaweed growth. But what I wanted to find out is, can this local cycling actually have an important effect? And so that's where I'm going to bring up these experimental designs. How do we partition the consumptive versus facilitative effect of these grazers? So I'm going to show you two different versions of a similar sort of design, where here is a container. We have snails. In this case, it's Littorina littorea, a snail species that lives on the east coast. Algae on both sides. Ammonium can diffuse across this mesh barrier. So you have ammonium concentrations relatively equivalent on both sides. These algae receive ammonium, but no grazing. These ones are being grazed and receive ammonium, and then you have treatments here that have no grazers present at all. And this actually ends up being a much more complete design for teasing out both the consumptive and non-consumptive effects of these herbivores on algae, because it allows us to make these comparisons and look at the mechanisms by which the grazers are affecting algal growth and productivity. So here, just to look at ammonium concentrations, this is the grazer present side, which I've abbreviated GP, the grazer barrier side, which is GB, and no grazer treatments. And you can see that ammonium concentrations are roughly similar in the grazer present and grazer barrier treatment, but they're lower if there's no grazer present. And so what I'm able to do is to partition the consumptive effect, which is grazer present minus grazer barrier. So the difference between these two, provided that the ammonium is diffusing across, is mediated just by the consumption associated with these grazers. A facilitative effect can be calculated by comparing this grazer barrier treatment that's receiving ammonium but no grazing with the no grazer treatment. And then a total effect is grazer present versus no grazer. Okay, so this is an example of how I did this first in the lab, but then I also did this in the field. So I'll show you an example using this design as well. So in this case, I used travertine tiles as a substrate where I can allow algae to recruit and grow in the field. And then I make fences around some of those tiles and not other tiles. And then grazers are manipulated at the scale of entire tide pools in the field. So there are grazer present versus grazer removed tide pools. And that allows me to use a similar sort of design to partition the consumptive versus non-consumptive effects of these grazers. Okay, so again, this is equivalent where you have grazer barrier treatments. So there's ammonium that's diffusing across and affecting the algae growing on these tiles, but no grazers. These are grazer present tiles, and then there are no grazer tiles. Okay, so now I want to highlight the importance of community composition and mediating these important effects that grazers have on both top-down and bottom-up effects on algal abundance. So one thing that you need to realize in thinking about interactions, especially in a field setting, is that grazers have a lot of different algal species to choose from. These are just some representative species that Littorina littorea on the east coast of the US feeds on in intertidal systems. And so we've sort of transformed this from here's one grazer, Littorina. It has a whole bunch of different algae to choose from, and we're going to try to partition its top-down negative effect versus its bottom-up positive effect on these different species. And if you give all of these species together, give the grazers a choice, you see a wide variety of different grazer impacts. So this is the milligrams of algae consumed per gram of snail biomass. In some cases, remember, they have a choice, so all of these are presented together. In some cases, they're not consuming very much algae at all. In some cases, they consume a lot, so Porphyra, I guess it's now Pyropia even there, and Ulva, they're consuming quite a bit. And then this one really struck me. So this is Fucus vesiculosus, and it actually has a negative grazing rate. In other words, in the presence of grazers, where there are other algae around, it actually grows better than it does if there are no grazers present. So there's a facilitative effect going on. And so because they were the extremes, here you have Ulva and Fucus, I focus specifically on that comparison. So you have a palatable species that's clearly being grazed a lot, and a species that there's some evidence for an actual positive effect of the grazers if they have a choice. So again, I used this design to evaluate how one seaweed species modifies the grazer effects on a second seaweed species. Remember, Ulva is highly palatable. Fucus may actually benefit from the grazers. And I'm using this design that I already described to you. Okay. It gets a little more complicated, so I combined an additive and replacement series design in conducting this experiment. And so what that meant is that I had a treatment that had low biomass Fucus, high biomass Fucus, so it has twice as abundant, low biomass Ulva, high biomass Ulva, and then Fucus plus Ulva. And that design is important because it allows me to avoid confounding seaweed density with community composition. Okay, and if you think about how to design experiments when you're looking at these types of interactions, that's pretty important. All right, so what did I find? So in these next couple figures, they're going to look something like this. The y-axis will be growth. In the first case, this is growth of Ulva as a percent of initial mass. And then what I've done here is partitioned these different effects. So these bars are comparing consumption, these ones facilitation, and these ones the total herbivore effect. The blue bar on the left is where Fucus is absent. The green bar on the right is where it's present. You can see that there are strong consumptive effects. So that Ulva is consumed a lot by Littorina, but that the presence or absence of Fucus actually modifies that. So when Fucus is present, they actually eat more Ulva than when it's absent. There's a negligible facilitative effect, and the total herbivore effect pretty much mirrors the consumptive effect. So in terms of ulva, consumption, top-down effects are what are most important. Now let's look at the effect on fucus. The first thing I want you to actually notice, in the case of ulva, this is a percent of initial mass and it's going all the way down to 90% effect. So these are really, really strong consumptive effects. If we look at fucus, the effects are much smaller. Remember, fucus is a less palatable species. What we see is that the snails eat fucus where ulva is absent, right here, but not where it's present, okay? We also see that when ulva is present, there's a positive facilitative effect of snails on fucus. And so what that translates to is in the presence of ulva, you have a slight negative effect, but not very strong, and actually a positive facilitative effect. And those together translate to a negligible total herbivore effect. But understanding these mechanisms is important because this negligible total herbivore effect emerges as the sum of a slight negative and a slight positive effect of the grazers on the alga. So again, we see evidence that there can be actually positive effects of grazers on seaweeds, and that the total herbivore effect is a combination of these top-down negative and bottom-up positive effects. So in terms of community composition and its importance, snails are clearly enhancing local scale nutrient availability. We see ammonium accumulating. And the effects of grazers on seaweeds and the effects of grazer-mediated nutrient availability on seaweeds depend on community context. Grazers can actually enhance the growth of these less palatable seaweeds, especially when a more palatable seaweed species is present. Okay, so this was done in laboratory microcosms, and now I want to move into the field and say, well, can we actually see similar things emerging under field conditions? Okay, so that's the final part of my talk here. When I was first interviewing for my job at UC Irvine, they took me down to Crystal Cove State Park to show me the cool intertidal spots, and I started poking my head in the tide pools, and this is actually a picture I took when I was on my interview. And you can see there are all kinds of cool things. There are chitons and a few different snail species. So there's littorines, and there's tegula, and limpets, and not much in the way of macroalgae. So I was like, all right, I'm going to have to figure out how to study this system, because there are a lot of grazers, not much macroalgae. What are we going to do here? Now, I actually don't do most of my work at Crystal Cove just because it's harder to get permitting in the state parks. I do my work at Corona Del Mar State Beach, which is great. It's about a 15-minute drive from the UCI campus. I can get a lot of undergrads involved in the research there. And as you can see, there are a lot of tide pools that act like really great little sort of field mesocosms to do my research there. And this picture here was actually taken in one of these tide pools, so you can see there are a lot of grazers, and they're actually grazing on the microalgae in these pools. Okay, so I wanted to study the effects of grazer abundance on nutrients and algae. So the first thing I did was surveyed tide pools for snail abundances. I also measured ammonium concentrations in the water, and then because I was dealing with microalgae, which are tougher to survey, I started using pamphloropmetry to actually quantify standing stock biomass and productivity and things like that. And so you can see here, this is dark adapted fluorescence as measured on the pamphlometer, and this is extracted chlorophyll, and we can get these nice positive relationships and get a non-destructive measure of standing stock algal biomass, or at least chlorophyll concentrations, in response to different experimental treatments and to the presence of grazers in the system. Okay, so this first slide shows the effects of grazer abundance on nutrient concentrations. So snail abundance is here on the x-axis, ammonium concentrations on the y-axis, and as snail abundances increase in these tide pools, we see a corresponding increase in ammonium concentrations. We also see a corresponding increase In this case, the y-axis is photosynthetic biomass, or the dark adapted F0 measurement from the PAM fluorometer. So as snail abundance increases, we also see an increase in photosynthetic biomass. Okay? So there are a couple different possibilities here. One is that the snails aren't just eating the algae, but they might help them by providing nutrients. The second is just that the snails are going where there's lots of food, right? Because they're really mobile, surprisingly mobile. I don't know if any of you have ever tried to maintain a snail removal treatment in tide pools, but as soon as you take them away, they go right back, okay? Over the scale of an area the size of this room, I think Tegula and other grazers basically roam with abandon at high tide. They're all over the place, okay? So I wanted to do an experiment and say, to actually try to establish whether this relationship was causal. So I manipulated grazer abundances in the tide pools. This is just sort of a schematic, right? Tide pools don't occur in a grid like that. The only place you find a grid like that is in Mike's seaweed farm. Here you have addition tide pools, controlled tide pools, and removal tide pools. What I did was I removed the grazers from the removal pools and added them to matched addition pools. And they were matched by volume, so that I was taking sort of an appropriate amount of grazers from one pool and then moving them into a matched similar type of tide pool. I also, in the control pools, picked up and dropped grazers just to control for that disturbance. Here you can see grazer abundances in these treatments. Like I said, it's hard to maintain, so they tend to go right back into the removal pool. So if I go back a few days later, they're right back in there. So I guess what you have to imagine is that a few days before I took this measurement, it was at 0, and so the actual value is somewhere in between here. Control and addition in terms of grazer abundance and number per meter squared. And then, so I'm manipulating grazer abundances at the scale of whole tide pools, and then within the tide pools, I manipulate grazer access. And so I have these experimental tiles, some that grazers can get to, and other ones that have fences around them that grazers can't get to. And so those look like this. We have fenced tiles and unfenced tiles. And if you actually look at them in the field, here you have an unfenced tile with a whole bunch of littorines crawling all over it, and a fenced tile that's pretty clean. Okay, and these fences are pretty effective as long as I go out there frequently and maintain the treatment. So here's the fence effect in the removal treatment, the control treatment, and the addition treatment. And the bottom line is that fencing is more important when there are more grazers. So here in the addition treatments, there's a much stronger fencing effect because there are more grazers present. Okay, so I have manipulations of grazer abundances in the tide pools at the scale of whole tide pools, manipulations of grazer access within the pools, and then I quantified the effects of those treatments on photosynthetic biomass over a month and a half to see what the trajectory looked like. And in fact, I'm going to report these data back calculated to chlorophyll A because that allows me to use different gain settings on the PAM fluorometer. And I have a good relationship for each of the gain settings between dark adapted fluorescence and chlorophyll A. So the values that I'm presenting are back calculated to chlorophyll A, so they can be on a consistent scale all the way through. These tiles, these are travertine tiles, which is a pretty good match for the Monterey formation of these intertidal reefs in terms of rock type. I anchored the tiles in the tide pools 2 months before the experiment to allow microalgae to recruit and grow on the tiles. And in this case, I'm going to use the chlorophyll concentration immediately prior to beginning the experiment as a covariate. And it turns out that pool dimensions, surface area to volume ratio, are also important in mediating microalgal growth because different pool size has implications for things like temperature and productivity. So I'm going to incorporate surface area to volume ratio as a covariate. Okay, so it turns out that both the initial chlorophyll concentration and the pool surface area to volume ratio are important. After accounting for those, the effect of herbivore exclusion, in this case the fences, depends on the grazer removal treatment. So here you have the grazer treatment, removal versus control versus addition, and this is the fence effect. So to calculate the fence effect, this is simply the chlorophyll concentration inside the fence minus the chlorophyll concentration outside the fence. A positive fence effect, then, is indicative of consumption. A negative fence effect is indicative of facilitation. And what we see is something really interesting, where in the removal treatments, if I've tried really hard to keep the grazers out of these tide pools, there's a positive fence effect, which means there's a consumptive effect of the grazers. But that changes along this gradient, so that by the time we get to these addition pools, there's actually a negative fence effect. So that means that chlorophyll concentrations are higher where the grazers have access to the tiles than they are where I have experimentally excluded the grazers. So in this case, the grazers are having a positive effect on algal abundance. So what's going on here? Well, one possibility is that these consumers do have effects on ammonium concentrations, and these could manifest at very small scales. So here, I've done measurements over even a single tile where you have grazers on one side and not on another, and there are some subtle differences in ammonium concentrations. So they're slightly higher on the snail side than the no snail side, but I really don't think that that's enough to explain these differences that I'm seeing. The second possibility, and this actually has a precedent in the literature, and I'll get there in a minute, is that there's the possibility that snail slime could be actually enhancing microalgal recruitment and growth. So as described in a reputable journal like The Guardian, snail slime actually does have important applications. It turns out it can be really important in mediating algal productivity as well. So this is a paper published in Science in the early 80s by Connor and Quinn, highlighting how limpet mucus stimulates the abundance of the food that they're eating. And they did these really great experiments up at Bodega where they got limpets to crawl around on things, and they put them out in the field, and they did all these measurements, and they actually showed very convincingly that the slime that these gastropods are laying down on the rock acts as an adhesive trap and promotes microalgal recruitment to the benthos. And I think that that's probably what's going on here. Here's one piece of evidence. I'm still working on this problem, and I'm trying to figure out how to get to collect mucus from various grazers and put it out in the field and finally show what's going on here. I do know that if you put, these are just tiles, and this is a tile that has snails on it versus a control tile, and they were just left out there, blank tiles, left out there for a week. I went back out and measured it, and the dark adapted fluorescence, the chlorophyll concentrations, are higher on the tiles that had snails than the tiles without. So there clearly is, and these were empty tiles, so there's more recruitment of algae onto these tiles where snails are present than to tiles that have no algae present, or no snails present. Okay, so what we see here is multiple potential mechanisms by which molluscan grazers could actually facilitate microalgae. The first, and I've spent most of my time talking about that, is this ammonium excretion. But that's only going to recycle things at the local scale. So if the snails are in a tide pool, they're going to eat the algae in that tide pool and convert the nitrogen that's in that algal tissue into ammonium. And so that's just local scale recycling. And I don't think that that can explain this positive relationship. But if we also add in this aspect of the snails laying down a trail on the benthos and that enhancing recruitment into the system, then that provides a spatial subsidy of material from outside the system that can then explain this positive relationship. So we have this slime that's mediating the recruitment, the inputs of pelagic microalgae onto the benthos. So these are like pennate diatoms that are in the water column looking for a place to land and end up on the substratum. So I've spent a lot of time recently trying to figure out how to quantify slime. I cannot believe I'm standing here saying this to you right now, right? The first thing I tried, and there's actually some really cool work in Europe that's also looked at this, and they use activated charcoal dust. And it's kind of like fingerprinting, right? The slime is sticky, And you can sort of see the ghost of it here. So this is where the snail crawled around, and you can see that it's a little bit darker because we dusted it with charcoal dust. But that's kind of subjective, and it's pretty difficult to compare this to controls. What we've been doing more recently is that Uda Passow and Alice Aldridge at UCSB use an alcyan blue dye technique. They use it to assay marine snow, which is also a acidic polysaccharide that's falling through the water column. But these assays were originally designed actually to stain for things like respiratory mucus. And they're really good at staining mucus glycoproteins. So we have modified this assay to quantify snail slime, which is pretty awesome. And we found that different grazer species are actually producing different quantities of slime. So here you have slime. This is the alcyan blue absorbance of turban snails, littorines, limpets, and chitons, these different grazer groups. So we can put them on a tile and let them crawl around and then assay how much slime they left on those tiles. And one of the cool things that's emerged from this is that some species that produce less slime, like turban snails, are excreting relatively more ammonium and vice versa. So you have things like littorine snails that are producing more slime, but that don't excrete as much ammonium. And so this actually shows the percent contribution of each of these different grazer groups to ammonium excretion in the white bars and a slime production in the gray bars. And so you can see that here you have turban snails, and they're disproportionately producing ammonium. And here you have littorines, and they're disproportionately producing slime. And so it may be that these grazers are actually complementary in terms of the roles they're providing. And grazer diversity may be important in mediating the amount of algal productivity in these pools. And so you have a system that's kind of like this, where you have snails like Chlorostoma tegula that are excreting ammonium, so they're responsible for this link, and other snails like Littorina that are more responsible for this link. All right, so what I hope I've shown you here is introduce you to some new possible effects of consumers in these systems by highlighting effects of consumers on algal growth and even on algal recruitment in these systems. What's next in terms of this research? So one of my current NSF grants is to actually repeat these types of experiments all up and down the coast. So I've been running experiments like this in Southern California at Corona del Mar. I have 2 sites down there, 2 sites on the Central Coast at Rancho Marino Reserve, and then 2 sites up at Bodega Marine Reserve. What we know so far is that as the abundance of grazers increases, regardless of location, we do find increases in dark adapted fluorescence, and we're trying to tease out the mechanisms and see whether or not these mechanisms are the same as you move along the coastline where we have these gradients in things like nutrients and temperature. And really try to tease out the role of internal processes, consumption and recycling, versus external processes where you have things like these pelagic autotrophs whose recruitment to the benthos may be mediated locally by herbivores, and the role of nitrate as it's changing along the coastline is differentially available in the nearshore ocean as an external subsidy versus ammonium internally. All right, so just a few final thoughts. So typical perspectives when we think about the roles of consumers in marine systems and in mediating marine ecosystem functioning, emphasize the consumption of producers by herbivores. But what I hope I've showed you here is that herbivores can also benefit producers by recycling nutrients or by enhancing recruitment, and that in some cases, these positive effects can outweigh the negative effects. So snails and other grazers don't just eat algae, but they also help them. And so when we go back to this question, what are the consequences of the loss of consumers for the functioning of marine ecosystems? These consequences aren't just mediated via trophic pathways, but there are really other important effects like nutrient recycling, like mediating recruitment, that are also potential roles for consumers in these systems, and we need to understand and appreciate those roles as well. And with that, I will finish and be glad to answer any questions. Thanks.
I'll let you direct the question asking, and I'll go ahead and start. So, cool. Cool, I like it. We're looking at trying to ask the functional role of consumers in the system and understanding the idea of the duality of positive and negative, possibly for any given herbivore, right? Not necessarily talking about predators yet, right? Predators on herbivores and their potential positive role to algae.
Right.
Also peeing.
They're also peeing, and you have an indirect effect because they're consuming the herbivores as well, right? So it's like a doubly positive effect.
Right. So I like this round. But can you pull up that fucus mesocosm slide? Because I just want to have it up there as I ask the question that once you threw that slide up, I couldn't stop thinking about it for all the rest of your slides. Okay. And you know what I'm talking about? Yeah. And I think the reason I'm asking this is that on some species, you're going to get a clear result in your mesocosm experiments. Yeah, that one right there. Yeah. Which is what you showed previously for the alga. Right? Now, I'm only interested here just to see, I'm only looking at the green bars, and I'm looking at this as the total herbivore effect is nil. And the reason is because the herbivores are eating as much as they're facilitating. Yep. I love it, right? So now let's just imagine that alga's in the system, and the only bars that are relevant to us on the planet are the green ones. If we take the herbivore out of the system, they eat less and they facilitate less. So they'll still have a zero net herbivore effect because they're not there. Right. So now I'm looking at like whether they're there or not, the effect is the same. Because what they remove, they facilitate. If they're not there to remove anything, they don't facilitate anything. So the net for fucus is they actually grow more, right? But it seems that the standing stock is the same because if there's no herbivore, they don't get to grow more to have more to eat. They grow less, so they have a lower standing stock. So then the question is, does the herbivore really matter? Because whether it's there in the first place or not, you still have a nil herbivore effect. Is it?
If there's alga there.
That's what I'm saying. Just in the world of alga being there, I totally understand how it changes when alga's out of that system. But in that consequence, right, the fact that the herbivores are facilitating and consuming at the same time potentially makes, in that context, their effect nil, whether they're there or not, because it's like, great, the alga grew, the fucus grew more, and that same amount that it grew got removed. So there's no net effect whether they're there, and there's no net effect whether they're absent.
For that one species.
Yeah, and then so I'm thinking this is neutral, right? When the same species creates a pot, I remember those diagrams where we talk about all the positives and negatives and commensalism and immensalism and all that. I'm finally seeing a neutral effect where when everything's working, nothing happens.
Which is kind of cool, right? Because it means it's stable.
Where it's cool to realize it's so damn complex that in the end nothing happens, and it is what it is, which I just think is cool. So you pull it apart, you put it back together, and you see nothing. Right?
Right. But if you look under the hood, there's all kinds of cool stuff going on.
Yeah. And then my grand comes in and takes on the alga, and the whole system goes back. Because the nice balance that was maintained in the presence of alga is no longer maintained.
Right. And so then you have these emergent strong consumptive.
Because their preferred food is no longer present, and the darn snail says, all right, you took the alga away, now I gotta eat this crappy brown alga, which turns out to respond pretty nicely to me being there in the first place. And so I get it. But I'm just wondering, the deeper you get, without looking under the hood, the more simpler it appears. So I wonder how the general public, sits back and views this, which they pretty much say, it doesn't matter if we take the snails out or not, I'm at. Right? If we lost this is Tegra.
This is Littorina littoria.
If we lost Littorina in these plots, but we didn't lose alga, you wouldn't notice the difference. On the fucus, right?
On the fucus. But you would notice a big difference for the alga.
You would.
Because if you go to that slide.
Yeah, you're right. You're looking at the whole darn system, and I'm just taking a little component of it. But in that very simplistic view, you can actually get to the point, and maybe that's the lesson, is that if you don't sit back and look at the whole system, you can get a completely wrong interpretation, right? Because you're not looking at how all these things are compared.
And you don't know the mechanism.
Yeah.
Have you ever looked at microalgal preference versus macroalgal preference for these consumers? Like, if you were to do the exact same experiment with and without the presence of microalgae and ulva, like, would there be a preference by the consumers to favor microalgal consumption versus the consuming ulva?
I think they would probably prefer microalgae, is my guess, just observing them. I mean, at that stage though, where they're grazing in things like tide pools, I'll be able to tell you more. I have a PhD student right now who's actually doing the genotyping. So she's using 16 and 18 sRNA to try to probe the community composition in response to these grazing experiments. And we'll be able to actually answer that question, sort of what's the composition in terms of, it's both prokaryotic and eukaryotic. There are all kinds of cool nitrogen transformations and things going on in the system. And the grazers are mediating that whole process. But we'll actually be able to tease out things like diatoms versus macroalgae at the micro scale and start to answer some of those sorts of questions. My guess, just from observing them, is that they would prefer microalgae, things like diatoms, to macroalgae, but I'm not sure. At the micro scale, in these pools where I'm looking at them, where they're basically equivalent in size. Once the macroalgae grow up, they may hit a size refuge, depending on the species. So that's the other thing to keep in mind. Because most of these grazers are they're rasping on the rocks, and that's where I think they get most of their food. In fact, I'm starting to lean toward the role of diatoms and other microalgae on the rocks in terms of conduits of energy into intertidal systems as being at least as important, if not more important, than the macroalgae that a lot of us have focused on for so many years. Yeah?
I was wondering what's known about the nutrient limitation, whether the benthic diatoms that are growing on these rocks in the tide pools, whether they become nutrient limited, and how does the supply of ammonium from the grazers compare to like the resupply of nitrogen from the ocean when the tide comes up?
So in both cases that I've shown here, so the case in New England and in Southern California, nutrients are pretty chronically low in the nearshore ocean. So nitrate plus nitrate concentrations in the seawater are probably at or less than 1 micromolar. And then a lot of, but nutrient dynamics inside the tide pools, concentrations can become a lot higher as they're mediated by consumers in the pools. So I think a tremendous amount of the nutrient supply is being controlled or mediated by the consumers in the system. I think if consumers are absent from the pools, then there's a really high potential for them to become, during low tide, for them to become nutrient limited, because you saw that the ammonium profile that I showed from the actual tide pools. It's clear that the microalgae are taking up a lot of ammonium when it's supplied to them. And so if it's at sort of ambient low levels, if there weren't any consumers contributing local scale nutrients, I think that they would very rapidly deplete the ammonium or nutrient concentrations in those pools, so that there would be very little left shortly after the pools were isolated.
These data are collected the data from Brenda Mark, which is most of what we did in the last Latin talk, is 2017, 2018 data. When did you first start collecting that data that made it into these graphs and vegetative fish from Brenda 7, 10 years, Southern California has a significant amount more of upwelling-driven nitrates.
Yeah, there's no upwelling in the system right now.
Yeah, so
it hasn't been for a while.
But it will come.
We hope.
And when it does, right?
That could reset the system.
Right, because now it's going to be independent of the Grazer component. Okay, because I'm thinking now you're doing the data.
Right.
Where it's pumping. And Rancho Marina would be much more so.
Yeah.
So you would imagine in that case that the competition for nitrate, or nitrogen, either through nitrate or ammonium being the species that the alga is preferring, right, will become a bigger part of the story as you move up that coast. I'm just assuming. Right, because our ammonium is minor relative to the amount of nitrate that's available. I mean, we're looking at 50, right now outside of Los Angeles, 13 micromolar
Of nitrate.
of nitrate.
Yeah. But in these tide pools, you can look at tide pools at Bodega, ammonium concentrations can rival or exceed nitrate concentrations there too. So actually the whole system is cranking higher. So we actually find ammonium concentrations and even phosphate concentrations. I've measured phosphate concentrations in these pools of like 60 micromolar.
I'm just wondering if facilitation becomes important when nitrogen becomes readily available. Will there be an ammonium effect when there's already more than enough nitrate to system?
So we did, Jay and Chris Aquilino and I did an experiment on that where we looked at pyropia in mussels, and we found that even there at Bodega, pyropia is getting a lot of its nitrogen from growing on mussel shells. Even on what can be arguably some of the most wave-swept shorelines in the world, where you have a ton of upwelling mediated nitrate coming through the system. Because ammonium is such a better source of nitrogen for seaweed.
I just remember a paper you wrote where you showed a graph of various species of algae where some assimilate and prefer nitrate over ammonium, right? And you argue that the diversity of the species of algae in the tide pool will vary. So I'm just thinking you might have some of these things like fucus that are much more on the nitrate level side of this, whereas ammonium and pyropia.
And pyropia is actually right in the middle. It can go either way.
All right.
And something like Clitophora or Ulva clearly prefers ammonia. So it really depends.
More? No? All right, excellent. Thank you.
All right, thank you.